Welcome to EV Club. That's co-hosted with GUSEV. We have some great talks for you today. And before we start, just a brief introduction about GUSEV. So we evolved from the ICEV Rigor and Standardization Urine Task Force with a focus on EV biology and function in the genitourinary system. Our focus is education, cross-disciplinary, cross-society collaboration, and professional networking. The QR code on the right will take you to our registration page. So we're very excited to announce the next GUSEV Education Day is going to be held on the 20th of February next year, and it'll be a virtual event. The theme is single EV analysis with nano flow cytometry from bench to bedside. We've got fantastic speakers and talks lined up. The GUSEV Education Day will also double as GUSEV's next EV club. And GUSEV will also have a satellite event, ICEV 2025 in Vienna, and we look forward to seeing you there. Also, GUSEV is recruiting, and we have a survey for you to let us know how you would like to engage with us and participate in our activities. The QR code will take you to our survey. And now, without further ado, I'll hand over to Dylan Berger, Cristina, Grange, who will moderate tonight's enlightening session. Thank you.
Thank you, Monica, for the introduction and for explaining the very nice activity of the GUSEV. So we are excited today to have all the three authors of the article present with us today. So let's get started with introducing our guest, Dr. Jannik Larsen. He concluded his PhD in 2024 in the University of Southern Denmark. He is now a postdoc working on approaches to determine AV dynamics in vivo. Then we have Dr. Iben Skog Jensen. She is an MD from the University of Denmark, and she is in the first year of the PhD. She is studying how proteinuria affect kidney physiology in the animal model and humans. And finally, we welcome the Professor Per Svenningsen, Professor of Kidney Physiology at the University of Southern Denmark. His group used EVs to understand the molecular physiology in the human kidney disease and physiology. So please go ahead. The stage is yours.
Thank you very much. I'll open my presentation here. Perfect. Yes, thank you very much for the kind introduction and also for allowing us to present our work here. So if we want to understand the function of extracellular vesicles and also use extracellular vesicles as more advanced biomarkers, it is important to know the cell type of origin of the EVs that are circulating both in plasma and those that we can extract from urine samples, for example. It's possible to track or identify the cell of origin used in different kinds of animal models. For example, I'll find my pointer. Yeah. So in animal models where you can use genetic labeling of cell type specific EVs and thereby create a robust link between the cell type and the circulating EVs. In humans, we have flow cytometry where we use affinity reagents like, for example, cell type specific antibodies to determine the cell type specific abundance of EVs in different biological fluids. But one of the main hurdles about the flow cytometry here is that we need these very specific affinity reagents, so cell type specific antibodies, and the number of cell type specific antibodies available at the moment is rather limited. So another approach would be nice, and one of the things we can do is to look at the RNA inside the extracellular vesicles, and then use what is called transcriptome deconvolution methods to estimate the cell type specific EV abundances in biological fluids. And most of the transcriptome deconvolution methods have been used and developed to estimate the cell type abundance from bulk RNA sequencing data of tissue samples. And if you boil it down to its core, what the deconvolution method does is to find a solution to this equation here. So T represents the bulk RNA sequencing data, so the abundance of the gene expression for a lot of different genes. And then you have S here, which is a signature matrix generated, for example, from a single cell RNA sequencing data, allowing us to create a sequence, a transcript profile for the different cell type. And what the deconvolution method then does is to find the solution here by estimating the cell type specific abundances here. And this works great for estimating cell type specific abundance in bulk RNA sequencing data generated from tissues, and it has also been used in a few cases to determine the cell specific EV abundance in body fluids using transcriptome data from EVs isolated from, for example, from blood and urine. But one of the assumptions that is made on the present transcriptome deconvolution method is that the RNA from the bulk RNA sequencing data and the RNA put into the signature matrix here comes from the same compartment, so more or less the whole cell. And we all know that EVs are more or less a separate compartment. So it remained a question whether these existing transcriptome deconvolution methods could reliably estimate the cell-specific EV abundance in these biofluids. So what we did was that we identified 11 different deconvolution methods here, and then we tested them through a panel of different tests. And what we started out by doing was to use RNA sequencing data from 4 different epithelial cell line and their corresponding extracellular vesicle. And then we used the deconvolution methods here to create a signature matrix with the cell RNA expression profile here, and then perform the deconvolution on the EV RNA expression profile. And if everything goes okay, then we should have a pattern like this diagonal here, which represents the true sample. So as you can see here, the DWLS method here and CyberSort X method here performed well, and created this nice diagonal pattern here, indicating that they could identify the cell of origin correctly. And you can also see it here more when we quantified the similarity, that these DWLS method and the cybersort method here produced estimates that were really close to the true sample. So in biological fluids, there's of course not only one cell type specific EV, one type of cell type specific EV present, but it's a mixture of different cell types derived EVs. And to find out if the deconvolution method were able to estimate correctly a mixture of cell type specific EVs, we used we used in silico mixtures of the RNA sequencing data, and using a very recently described method. And in essence, what we did was that we took the RNA sequencing data and then randomly extracted a subset of reads from these two cell type specific EVs here, and then mixed them into 5 different fractions, so that we had between 0, 25, 50, 75, and 100% of cell specific EVs present in these 5 fractions. And here is the result shown for the DWLS based deconvolution, showing that the DU 145 samples here showed that the estimate of the abundance was highly correlated to the mixing ratio here. And also when we tried to decrease the abundance of the DU 145 derived EVs in the mixture, there was a nice correlation to the mixing ratio here. And you can see there is maybe a slight overestimation here at the very low abundant mixtures here. So when we compared the 11 different deconvolution methods here, and compared the different metrics here to quantify their performance, you can see here again that the DWLS method and CyberSort X performed reasonably well. We also went on to test mixture again, using this in silico mixture approach, but this time we used 3 different samples. So we created 12 different in silico mixtures with 0, 12.5%, 20, 25, 30, 37.5%, and 50% of cell-specific EVs. And you can see the true pattern is the panel here on the left, and then you can see the performance of the different deconvolution methods to the right here. And again, you can see that the DWLS method here and CyberSort perform really close to the true mixing sample here. And importantly, the amount of this CRC EV is really low in these sample here. These method here are highly consistent and very robust. Another way to test the robustness here, the deconvolution method that we included here were all marker based, meaning that we can only detect the EVs which we also have markup for in the signature matrix. And this also means that EV RNA from cell types not present in the signature matrix could interfere with the estimates. So to test how robust these methods were to the introduction of foreign EV RNA, we mixed this DU 145 EV RNA samples with adipocyte and macrophage EV RNA sequencing reads, and produced four different samples here, consisting of 0, 25, 50%, and 75% of this adipocyte and macrophage derived EVs. And you can see here for the DWLS estimates here, is not significantly affected by the introduction of these adipocyte and macrophage derived EV sample, and the same goes for the CyberSort X method here. So on the in silico mixtures here, we quantified 14 different parameters, and then we included this in this graph here. And the closer to bullseye it is, the closer to zero it is, the better the performance of the deconvolution method is here. And as I've said a few times now, the DWLS method and CyberSort X performed really well on these in silico mixtures here. So we went on to test whether these four methods here, which are highly consistent on in silico mixtures, also produced similar estimates on real samples from humans. And to start out, we used urine samples obtained or urine extracellular vesicle RNA sequencing data from two different cohorts, and tried to estimate the tissue and cell type specific abundance of EV in these samples. And to start out, we used a tissue matrix consisting of 27 different tissues obtained by getting RNA sequencing data from GTEx. And here is only shown the DWLS estimates for the two different cohorts. And what is striking here is that, for example, the pancreas, skin, and also stomach derived EVs appear to be highly abundant. And this conflicts somewhat with other experimental data, which suggests that urinary EVs are mainly coming from the genitourinary system. And this huge or this 27 tissue type signature matrix also produced estimates with the different kinds of deconvolution methods that were not that well correlated. So because pancreas and also stomach here were highly abundant, we we thought that maybe it was the epithelial cells in these organs that somehow could interfere with the deconvolution. So we instead created a restricted tissue signature matrix consisting only of tissues in the genitourinary system. And as you can see here in panel B, using this restricted tissue signature matrix created estimates that were highly correlated between these four high-performing deconvolution methods in both our urine cohorts. And what this suggests is that the kidney and bladder derived EVs are abundantly present in urine samples. And also we found that the estimates here is shown for DWLLS, that the estimate from this deconvolution method was highly similar between these two urine EV cohorts here. So we went on and also wanted to test the cell-specific origin of the EVs, and to this end we used a single-cell RNA sequencing data set of human kidneys, and included a signature matrix consisted of proximal tubule cells in the collecting duct, and the DCT, and also in the loop of Henle and in the connecting tubule. And what we found was that proximal tubular cell-derived EVs were highly abundant in the urine sample, and then the collecting duct, principal cell and intercalated cell were also present at a fairly high number, while the other cell type derived EVs were present, but at a much lower level. And again, you can see here for the DWLS method here that the estimates is highly consistent between these two cohorts here. So this was the urine samples. Then we also wanted to test in plasma EVs, so EVs isolated from plasma samples, and we found the RNA sequencing data set of plasma EVs. And what we found was that the best performing deconvolution methods here predicted that most of the plasma derived EVs were either from blood or from spleen here. And spleen, of course, contains a lot of the blood cells, so immune cells and so on. So we combined these two compartments here, and then compared these four different, the four best performing deconvolution methods estimates. So the estimate from the DWLS showed really high correlation and similar estimates to the other DWLS based methods, and also to CyberSort X. And again, we wanted to test the cell-specific origin in a subset of these blood cells, and more or less all of the deconvolution methods here estimated that monocyte-derived EVs were the most abundant EVs in plasma. So together, these results indicated that it is possible to perform a reliable deconvolution of EV RNA to infer their cell type specific origin. And what can this be used for? So one of the things is that this can be used to monitor the appearance or disappearance of cell types. So, for example, in the treatment of a cancer, you can measure whether or not a treatment is successful by monitoring the disappearance of this cell type specific EVs. It could also be used to normalize other kinds of omic data, so that you can correct for changes in cell type specific EV abundance in proteomic and lipidomics EV data. And it can also be used to monitor dynamic changes in EV abundance. And I'll just draw your attention to this graph that I have shown you before, where we saw that the proximal tubular-derived EVs appear to be most abundant in urine samples, while integrated cells of the collecting duct were less abundant. But if you normalize this to the number of proximal tubular cells and integrated cells in a normal human kidney, you will find that the ratio between integrated cell-derived EVs and the integrated cell number is roughly 5 times higher than the number of proximal tubular cell-derived EVs to proximal tubular cells. So this could maybe indicate that this method could also be used to estimate the cellular secretion rate, at least in acute conditions where there haven't been any structural changes to the kidney, and maybe expansion of certain cell types. Yeah. So it's not that easy to at least in chronic conditions to infer the cellular EV secretion rate from these data here. But maybe another approach could be used, and I just want to draw your attention to this paper here, where they show that it's possible to actually extract the number of extracellular vesicles derived from single cell types using a single cell RNA sequencing data, and this method could maybe be used in future studies to also, on the more chronic conditions, investigate EV secretion rate. Of course, with the caveat that this is a highly invasive procedure where you will require a sample of the tissue. So, nonetheless, the conclusion of our paper here is that the transcriptome deconvolution here provides an approach to estimate the cell type specific EV abundances in biological fluids, and we have to note that our benchmarking study is based on in silico mixtures, and one of the assumptions here is, of course, that the RNA content in EVs is similar between different cell types. We don't know that if that is true at present, and therefore it will also be nice to see some real world mixed EV samples. And another thing is that as I just said that the deconvolution methods that we used here are marker based, so we need to include cell markers, and it could also be important to develop this field further, and maybe implement some techniques where we could deconvolute the cell of origin without the need for these cell type specific markers, and also detect if unknown EVs are present. But our recommendation based on our study here is that the DWLS and CyberSort-X methods here provide reliable abundance estimates. And the nice thing about this is that these methods, they use two different approaches to create their estimates. And then, as we have seen, cellular subtypes and tissue with similar cell type composition might bias the deconvolution. And one way to mitigate this problem is to build a signature matrix here with biological knowledge in mind. So different and anatomical restriction could be included in the design of the signature matrix. And with that, I would like to thank you all for listening, and I'm happy to take questions.
Thank you, Per, and congratulations to you, Yannick, and even for the great study and this very, very intriguing data. I think that's an inspired way to come from bulk data to a better knowledge of which single EVs or from which cell types the single EVs might stem from. Hello from Hamburg. Fabian Braun is my name. I'm the second moderator of the session, and I will now give you the chance and lead you a little bit to the question and answer section. If you want to ask a question directly, then please raise your hand, and then Dylan will, raise your hand digitally through the reaction button, and then Dylan will unmute you. I have 5 questions in the chat already, and I will start out with these. Per, Eben, and Yannick, Hong Zhang asks, are we using mRNA signatures for deconvolution in the cell and eVRNA analysis? And would it be able, or do you already include other RNA species?
Yeah, good question. We only used mRNA, but I guess everything that is cell specific could be included in the signature matrix. So yeah.
Maybe one additional housekeeping thing. If you have a camera and want to discuss, then please switch on your camera. It's always nicer to see people's faces than just the names. Second question from Maja. I hope I'm pronouncing that right. How many GTEx tissues were used as a reference? At the time of writing the Dwivedi et al. Paper, we found only 9 male bladder tissues, and we could not include it in the analysis due to such a low number.
I cannot remember the exact number. We have it in the table. We restricted an upper number, so we only restricted at 200 samples, but some of them were much lower in n numbers, and the bladder could be 9. It's in the paper, but I cannot remember it at the top of my head.
Thank you. Next question from Yetem Amsalu. Sorry if I'm butchering your names, German pronunciation here. Did you test the method for diseased and undiseased cell sources, or from healthy ones only? We know that EVs produced from a single cell are even heterogeneous, and this can be even more in healthy and diseased states.
Yeah, a great point. So at least the in vitro data, so the in silico mixtures are from healthy cells. For the GTEx, we it seems we use samples where we in the text the what's it called? They categorized by the way that the patients died. So we only included patient where there was acute death, not death due to long-term illness. So it represents more or less healthy tissues here.
Getting a little bit more towards a kidney-specific question, any idea why intercalated cells and principal cells would have a greater EV production rate compared to proximal tubular cells? That's from John Bisler. And I, as a podocyte researcher, would like to add PC stands for podocytes in the graph. They also had, if you compare that to the amount of cells, a quite high number then of EVs detectable, right?
So PC is principal cells in the graph, and we did not include podocytes. I cannot remember the reason why. We should have. Yeah, they are. So the cells in the collecting ducts are very metabolic active, and this could maybe be a response to being very metabolic active, that there's a higher secretion rate of EVs. We don't know that yet, whether this is the case, but this is our hypothesis. What we would love to do is to do RNA sequencing and deconvolution on samples from healthy humans on a high and low sodium diet to see if there's a difference in, for example, principal cell-derived EV abundance.
Maybe as a thought, could it also be that you find in total then less to the cell type because the EVs produced by the proximal tubular cells are taken up by cells down the nephron?
Yeah, that could indeed be the case also, and that's if any of you have a good idea how to control for this, that would be really nice to know, because at the top of my mind I cannot figure it out how to do it.
Next question is from Dylan. When you did plasma analysis, did you assume that the best performing deconvolution methods would be the same? Or another way, for saliva or cerebrospinal fluid, for example, would one need to go through a fresh benchmarking exercise?
I think they should perform similarly. So all the tests that we have put these methods through, it appears that CyberSort X and DWLS performs similarly. Yes, so I wouldn't expect it to be different in other biological fluids. There could, of course, be a case with the uptake of cell-specific EVs. So if some cell-derived EVs are engulfed by macrophages, for example, preferentially, then you would see a difference here.
And kind of in the same vein, but not from tissue, but from different cohorts, question from Cristina Grange. Do you think the deconvolution method needs to be adjusted when using cohorts of patient samples compared with cohort of healthy subjects?
Yeah, I think so. So yeah, we were mostly interested in both in the healthy state to just see if it would work. But I think that I think you would get a poor estimate if you use the signature from a healthy kidney and then use the urine sample from a patient with a chronic kidney disease or proteinuria or something like that. So the more similar the signature matrix and your EV sample can get, I guess, the better the estimates will be. And this can of course be a major challenge to find a signature matrix from a specific kidney disease.
Thank you. These were the questions in the chat. Do we have further questions from the audience? That does not seem to be the case at the moment. So, Per, thank you again for presenting. Once again, congratulations on the paper. Thank you everyone for joining. This was the last EV Club of this year. For every one of you celebrating any holiday now that the years are changing, happy holidays to you. Get safe into 2025 and see you next year.
Bye, everybody.